Good evening, everyone. Let us talk one more topic under biochemistry, that is ketone bodies. So, what are the objectives of our presentation? We have to define what is ketone bodies and what is the use of citric acid cycle and formation of ketone bodies, utilization of ketone bodies, identify significance of ketone body formation. Ketone bodies. The acetyl-CoA formed in fatty acid oxidation enters the citric acid cycle only if fat and carbohydrate degradation is appropriately balanced. So what they are telling here, acetyl coenzyme. It is formed in fatty acid oxidation. So oxidation, so any process is occurring under the presence of oxygen. Oxidation enters the citric acid cycle. Only fat and carbohydrate degradation is appropriately balanced. So what is the meaning of here? The fatty acid oxidation. It is a mitochondrial aerobic process of breaking down a fatty acid into acetyl-CoA units. So once again we can repeat. What do you mean by fatty acid oxidation? It is the mitochondrial aerobic process of breaking down a fatty acid into acetyl coenzyme units. However, if fat breakdown predominates, acetyl CoA enter into the liver undergoes different fate. So whatever is happening here, if fat breakdown predominates, fat is breaking down, acetyl CoA in the liver undergoes different states. So let us see briefly regarding what is citric acid cycle. The citric acid cycle is the first part of the third stage of food catabolism, which includes carbohydrates, fats and proteins. So what do you mean by citric acid cycle? It is the first part of the third stage of the catabolism, which includes carbohydrates, fats and proteins. So the meaning of catabolism, breakdown of complex molecules in living organism to form simpler one. Catabolism meaning is breakdown of complex molecules in living organism to form simpler one. The reason is that the entry of acetyl coenzyme into citric acid cycle depends on the availability of oxaloacetate for the formation of citrate. So what is the meaning of here? The reason is that the entry of acetyl coenzyme into citric acid cycle depends on. So how there is a dependency? The entry of acetyl coenzyme into citric acid cycle, which depends on the availability of oxaloacetate for the formation of citrate. Oxaloacetate is normally formed from pyruvate, the product of glucose degradation in glycolysis. So what is the importance of your oxaloacetate? For example, the acetyl coenzyme need to enter into the citric acid cycle, which depends on the availability of what should be available here? Oxaloacetate. Why it has to be availability? For the formation of citrate. So oxaloacetate is normally formed from how it is generally the oxaloacetate how it is forming? It is forming from pyruvate, the product of glucose degradation in glycolysis. So what do you mean by glycolysis? Breakdown of glucose to produce energy. Lysis, always remember this, the word is lysis is indicating breakdown. Then what is the meaning of your citrate? It is an organic carbolic acid and can be extracted from the juices of the citrus fluid. For example, lemon. If carbohydrate is unavailable, certain condition carbohydrate is unavailable, the concentration of oxaloacetate is lowered and acetyl coenzyme cannot enter the citric acid cycle. For example, this acetyl coenzyme have to enter the citric acid cycle which require oxaloacetate. This is the basic concept. If carbohydrate is unavailable, the concentration of oxaloacetate is lowered and acetyl coenzyme cannot enter the citric acid cycle. And once again, we can see here what is the meaning of this glycolysis? Breakdown of glucose to produce energy. Breakdown of glucose to produce energy. Then in fasting or in diabetes. So what is the condition? In fasting condition or in diabetes, oxaloacetate is used to form glucose by gluconeogenic pathway. So what is the what we need to understand here? In fasting condition or in diabetic condition, oxaloacetate is using to form glucose by gluconeogenic pathway, and hence it is unavailable for the first step of the citric acid cycle. So we have to remember this condensation of oxaloacetate with acetyl CoA. So what do you mean by condensation? It is a process where water vapor becomes liquid. Under these conditions, acetyl CoA undergoes to the formation of acetoacetate and beta hydroxybutyrate. Under these conditions, this like acetyl coenzyme which undergoes for the formation of acetoacetate and beta hydroxybutyrate. And let us see what are the types of ketone bodies. There are mainly three types of ketone bodies. What are they? Acetoacetate, beta hydroxybutyrate, and acetone. These three substances are collectively known as ketone bodies. So let us repeat once again. What are the ketone bodies? There are mainly three types of ketone bodies are present in our body. What are they? Acetoacetate, beta hydroxybutyrate, and acetone. These three substances are collectively known as ketone bodies. Acetoacetate and acetone are true ketone bodies. So what are the true ketone bodies? Acetoacetate and acetone are true ketones, while beta hydroxybutyrate does not possess a ketone group. The term bodies is an histological tag. Then these are water soluble energy yielding substances. These ketone bodies are what kind of substances? Water soluble energy yielding substances. Acetone is, however, an exception since it cannot be metabolized and is readily exhaled through lungs. So what is happening to the acetone? Acetone is, however, an exception since it cannot be metabolized. This acetone cannot be metabolized and is readily exhaled through lungs. Acetone is a waste product as it is volatile. It is, the meaning is evaporation. So automatically it may be exhaled through the lungs. It is identifying as a volatile state and it will be evaporate easily from the lungs. Acetoacetate and beta hydroxybutyrate are interconverted by mitochondrial enzyme that is known as beta hydroxybutyrate dehydrogenase. So what is happening here? This acetoacetate and beta hydroxybutyrate are interconverted. Both are getting interconverted by the enzyme name is mitochondrial enzyme that is known as beta hydroxybutyrate dehydrogenase. So acetoacetate continually undergoes spontaneous non-enzymatic decarboxylation to yield acetone. So what is happening? This acetoacetate prolongingly or continually undergoes spontaneous non-enzymatic decarboxylation. It is a mini spontaneous. It goes spontaneous non-enzymatic decarboxylation. What is going to yield here? Acetone. Then the concentration of total ketone bodies in the blood of well-fed condition. Well-fed means not in the fasting condition. The person had good food. The concentration of ketone bodies in the blood of well-fed condition does not normally exceed 0.2 millimole per liter. Then how the ketone bodies are going to be formed? Formation of ketone bodies occur in the liver. So where it is forming? It is forming in the liver and transferred to other organs as fluid. In some cases, these ketone bodies are helpful for the energy and these ketone bodies giving energy to the organs, or it is helpful for as a fuel to our body. So enzymes responsible for ketone body formation are associated mainly with the mitochondria. So what we need to understand here? So what are the enzymes? Some of the enzymes are responsible for ketone body formation or associated mainly with the mitochondria. Acetoacetate, beta hydroxybutyrate, and acetone are formed from acetyl-CoA. So how these ketone bodies are utilized by our body? The site of production of ketone bodies is exclusively livers. Where it is producing? These ketone bodies are producing in the liver. But the liver cannot utilize ketone bodies because it lacks a particular enzyme. The enzyme known as CoA transferase, and that is required for the activation of ketone bodies. Basically, the ketone bodies are producing in the liver, but liver cannot utilize these ketone bodies because lack of enzyme that is known as CoA transferase that is required for the activation of ketone bodies. Then acetoacetate, beta hydroxybutyrate and acetone diffuse from the liver mitochondria into the blood. How it is diffusing? It is diffusing from the liver mitochondria into the blood and are transported to the peripheral tissues. In peripheral tissues, the beta hydroxybutyrate is converted to acetoacetate and the acetoacetate is then activated to acetyl-CoA that is then cleaved to yield acetyl-CoA, which can be oxidized in the citric acid cycle to water and carbon dioxide. So let us repeat once again the sentence here. The peripheral tissues, the beta hydroxybutyrate is converted. In peripheral tissue, what is going to converted? This beta hydroxybutyrate is converted to acetoacetate. And the acetoacetate is then activated to acetyl-CoA. Then it is cleaved to yield acetyl-CoA, which can be oxidized in the citric acid cycle to water and carbon dioxide. So what is the significance of ketone body formation? Why the ketone bodies are going to be produced in the body? The production and transfer of ketone bodies from the liver to extrahepatic tissue. So what do you mean by extrahepatic tissue? Extrahepatic tissue means apart liver. Allows continued oxidation of increasing quantities of fatty acids in the liver when acetyl-CoA is not being oxidized in the citric acid cycle. So what is happening here? The production and transfer of ketone bodies from the liver to the extrahepatic tissues, which allows continued oxidation of increasing quantities of fatty acid in the liver when acetyl-CoA is not being oxidized in the citric acid cycle. During starvation. So what do you mean by starvation? Maybe like fasting condition. Intermediate of citric acid cycle are being drained off for glucose synthesis by gluconeogenesis, slows oxidation of acetyl-CoA by citric acid cycle, diverting them to ketone body formation. So what is happening here? During starvation. Starvation means fasting condition. Intermediate of citric acid cycle are being drained off for glucose synthesis by gluconeogenesis, slows oxidation of acetyl-CoA by citric acid cycle, diverting them to ketone body formation. Then during deprivation of carbohydrates, in the condition of deprivation of carbohydrate, as in starvation, like starvation and diabetes mellitus, in these two conditions, what is the first condition? In the starvation as well as diabetes mellitus. which is associate milleters, acetoacetate and beta hydroxybutyrate as an alternative source of energy for extrahepatic tissues as skeletal muscle, heart muscle, and renal cortex. So in the two conditions, so what are the two conditions? One is starvation, another one is a diabetes mellitus. In these conditions, what is going to happen? Acetoacetate and beta hydroxybutyrate is an alternative source of energy. These two are, what are they? Acetoacetate and beta hydroxybutyrate as an alternative source of energy for extrahepatic tissue as skeletal muscles, heart muscles, and renal cortex. And prolonged starvation. So starvation is will prolong time. 75% of the energy needs of the brain are supplied by the ketone bodies. When the person is having like a prolonged starvation, 75% of energy needs, our body energy needs of the brain are supplied by the ketone bodies, reducing its needs for glucose. These ketone bodies can be regarded as water soluble transportable form of derivatives of acetyl CoA. So how it was treated? These ketone bodies can be regarded as these are belongs to water soluble transportable form of derivatives of acetyl CoA. Therefore, they do not need to be incorporated into lipoproteins or carried by albumins and do the and as do the other lipids. So therefore, they do not need to be incorporated into lipoproteins. They may not, they don't need to be incorporated into lipoproteins or carried by albumins as do the other lipids. Then acetoacetate also has a regulatory role in lipid metabolism. This acetoacetate is also playing a role in the regulatory role in the lipid metabolism. High levels of acetoacetate in the blood specify an abundance of acetyl units and lead to a decrease in the rate of lipolysis in adipose tissue. Once again, repeat the sentence. High levels of acetoacetate in the blood specify an abundance of acetyl units and lead to a decrease in the rate of lipolysis in adipose tissue. So, what do you mean by lipolysis? Breakdown of lipids to produce energy. So let us summarize our topic. Till now we have discussed regarding ketone bodies. What is the definition we identified here, and so how these ketone bodies helpful us? Sometimes these ketone bodies is helpful for in the manner of energy producing bodies, and this is very important that citric acid cycle. So for the completion of citric acid cycle there is a formation of ketone bodies are necessary, and then we have talked about regarding formation of how the ketone bodies is going to be forming, and we discussed regarding utilization of ketone bodies, and finally we have spoken regarding the significance of ketone body formation. I hope my dear students you understand the concept of ketone bodies. Have a nice day. Once again we'll meet you.